Right. Hello. Good evening, students. How are you? Komada. Sorry, there was a computer crash, so I had to restart a couple of times. Komada, can you hear me clearly? Hope you can hear me. Am I audible? Am I Henawada? Yes. Okay. We'll go on marking today's paper. People, BioEve 8. BioEve 8. Find that paper. I gave that in the class. If anyone wants, please collect it from the chat line. Right, paper is in the chat line. You can download it. Then today's discussion, paper 8. Check this paper. Right, guys, I want you to mark the first few questions. Let me minimize it a little bit. Right, can you mark first 5 questions, people? Can you mark first 5 questions? Which of the following is a branch storage polysaccharide? Come on, mark it. Right. Some people have even marked different answers. Yeah, good. Right. So hemicellulose can't be neither. Hemicellulose can't be. Why people? They are storage. Right. Not only branched one. So answer is second one. Correct. Question number 2. Select the correct statement regarding lipids. What is your answer? Correct statement regarding lipids. They are most abundant group of organic compounds on Earth. No, wrong. Large biological macromolecules. No, they are not macromolecules. Large molecules. Large biological molecules, okay, but you can't say that they are macromolecules, right? Macromolecule means the molecular weight has to be greater than 10 to the power 10, 10 to the power 4 to 10 to the power 10, right? Above 4,000, sorry, above 10,000. So 2:1 ratio, no. Fat phospholipids and steroids are considered as biologically important type of lipids. Ek hari. Fatty acid molecules bind to each hydroxyl group of glycerol and phospholipid bonds. Fatty acid molecule bind to each hydroxyl part of glycerol by phosphodiester bond. Wrong, ne? The phosphodiester wrong, ne? So fourth one is the answer. Very good. Question number 3. Which of the following statement regarding structure of protein is true? Structure of protein is true. What is your answer for question number 3? Hydrogen bonds are required for formation of primary structure. No, wrong neither. Primary structure has peptide bonds. First is wrong. Namai, don't make big mistakes. Please study and answer properly. Hydrogen bonds are not present. Peptide bonds are there. Ne? Secondary structure is maintained by hydrogen bonds between R groups. Secondary structure is maintained by hydrogen bonds between. No, wrong neither. Secondary structure is due to hydrogen bonds between distant amino acids. No, the primary structure is stabilizing by hydrogen bonds. Primary structure is stabilizing by hydrogen bonds. No, peptide bonds. Formation of secondary structure require formation of hydrogen bonds, peptide bonds, sulfide. No, and therefore stability of the secondary structure is maintained by hydrogen bonds. That is correct, people. I'm taking fifth one as the answer for question number 3. Good. Question number 4. Which of the following is most similar in structure to ATP? Similar in structure to ATP. Why didn't you mark that one? Why? What's wrong? Dineth is marking an answer now. Pentose sugar? No, it can't be pentose sugar. No. DNA nucleotide, no. RNA nucleotide, correct. Amino acid with 3 phosphate group, wrong. And a phospholipid, no. So my answer is third one. Why do you say amino acid with 3 phosphate group? Nucleotide is a amino acid, wrong. Nucleotide is a nitrogenous base with 3 phosphate group and a sugar. So wrong. Question number 5. Select the correct statement regarding major component of cell membrane. It consists of 3 ester bonds. It consists of 3 ester bonds, cell membrane. Cell membrane regarding major component of cell membrane. Phospholipids, 3 ester bonds. Okay, now the first statement is doubtful for me because ester bond is a acid and alcohol interaction, correct? Fatty acids are saturated fatty acids, not always, neither. And additional polar molecule or small charge molecule is always linked, always. Wrong, neither, sometimes, ne? Can't say always, no? Wrong. Then one fatty acid molecule is replaced by phosphate group, correct? Hydrophobic tail consists of 2 fatty acid molecules. No, hydrophobic tail has hydrocarbon. Wrong. Not fatty acids. Keep that in mind. They are hydrocarbon tails, not fatty acid tails. No. So I'm taking fourth one as the answer. My answer is fourth one. One fatty acid molecule is replaced with a phosphate group. Correct. That is how it forms phospholipids. So I'm moving to next question. Question number 6. I'll clear your answers. Right, can you answer the sixth question now? Select the correct statement regarding scanning electron microscope. It is used to study internal structure of the cell. Wrong. A beam of electron is passing through. Wrong. Specimen are stained with dyes. Not wrong. Stained with dye. Ne? Pattern of electron passed through the specimen displayed on the screen. Passed through the specimen. Wrong. Important to observe three-dimensional view of the surface. Correct. I'm having two answers, third and fifth. Third and fifth. Two answers are there. Right, my friends. Friends. All right. So I got two answers here. My dear friends. My dear friends, we are moving to question number 7. Can you all give us small support for this part too? Yes. Question number 7. The main difference of TEM and SCM is TEM and SCM. What's the answer, people? Yeah, I think most quoted with gold, TEM wrong. Neither. We said SCM is gold. No, first is wrong. Beam of electron passed through the specimen, correct. Refracted, reflected from the surface of the specimen, fine. Beam of electrons reflected from the surface. Reflected from the surface, these scattered electrons are captured. Ne? That is what we said. More electrons may get displayed in the region where the structures were densely stained. And then specimen scatters many electrons, whereas others are absorbed. How about the third one? More electrons get displayed in the regions where structures were densely placed. Thin specimen scatters many electrons. No, the specimen scatters many electrons where as the others are absorbed. Did we say that it get absorbed? Scan electron microscope, did we say that it absorbs? I don't think we use that term. Ne? Absorption of electrons by SCM. Did you say that? Here the specimen scatters many electrons where whereas the others are absorbed. Mentioned. Ne? Specimen is mostly coated with gold prior to observation. Specimen is ideal to observe the surface view of three-dimensional in appearance. TEM. Image reflects the pattern of electron passed through the specimen displayed on the screen. While electron passed through the specimen, more electrons may get displaced in region where the structures were densely stained. More electrons may get displaced. May get displaced in regions where structures were densely. We had two answers now. I think we had two answers. A beam of electron is passed through the thin specimen. Yeah. So I'll go with second and third, Lamai. Agree? Do you agree with me? It is not mentioned specially prepared specimen. Beam of electron passed through the thin specimen. Why do you want me to say? Specially prepared slice of material. Beam of electrons passed through a thin specially prepared slice of material. Thin specimen. Thin specially prepared specimen. Yeah, there is a small weakness there, right? They have not, so it is not given like in the, so it is not given like that in the resource book. All right. But anyway, people, I don't see that the second one is that wrong. More electrons may get displaced. More electrons may get displaced. Yeah, we'll go with either second or third, then third best. Huh? Third best. I'll take it like that. Third best. Right, question number 8. Difference of prokaryotes when they are compared with eukaryote is a prokaryote show, binary fixation. Show binary fission. I think fourth one is the best answer. Fourth is best. Binary fission can be taken to. Binary fission can be taken to, right? First and fourth. Fourth is best. Some features of living cells are given below. Then what is the answer? B, C, E only. Which of the above features can be seen in prokaryotic cells? Can be seen in prokaryotic. Mitotic cell division, no. Mitotic cell division, no. Presence of 70S ribosomes, features living, can be seen in prokaryotic, present. Presence of subcellular components, present. And then presence of several kinds of RNA polymerase. That's the question, the doubt. Sometimes present, sometimes not. And you haven't learned that yet. Presence of polysaccharides and proteins in the cell wall. So we'll take fourth one as the best. Fourth one is the best. Right, next one. Question number 10. Select the correct statement regarding Golgi apparatus. What is your answer? Fourth one. Produces lysosomes. Correct. Fourth one is the answer. I'll take your answer. Computer stucks people today. I don't know why. I don't know why. Stucks. Right, moving. Oops. Oh. Right. 11, 12, 11, 12, 13, 14. Can you? It is given lysosome. Is that true? Lysosome. Which of the following organelle is abundant in liver for processing and packaging? Wait, previous one, produces lysozyme, wrong. Lysozyme is an enzyme, ne? Lysosome should be. Select the correct statement regarding Golgi apparatus. It says lysozymes, wrong, ne? I'm wrong. Right. 10th question, check it again. Golgi apparatus, it produces lysozyme. Inner and outer membranes are identified as trans and wrong. Transphase is located in the endodermic endoplasmic, wrong. Cisphase is facing towards the endoplasmic reticulum, right? Wrong. Secretory vesicles are budded off. Yeah, third one is the answer then. Wait. Secretory vesicles are budded off from the transphase. Cis trans, correct, neither. Third one. Golgi apparatus. Collecting, modifying, packaging, and distribution of carbohydrates are done by this. Secretory vesicles are budded off from the transphase. Produces lysozymes, wrong. Collect, modifying, packaging, and distribution of carbohydrate. Cellulose and non-cellulose components. Collecting, how it is given? Golgi apparatus. Manufacturing cellulose and non-cellulose. Collecting, modifying, packaging, distribution of materials, proteins and membrane lipids. Wrong, neither. So the answer is third. Third is okay. Can't go for 5. Third. Third one is the answer, people. Right, next one, question number 11. 11, what is your answer? 11, which of the following cell organelle is abundant in liver cells for producing packaging? I'm taking Golgi. Nee, that's correct. Nee, Golgi. Third, Golgi. Question number 12, which of the following correctly indicate the order of the structures when ammonium ion move from soil into the vacuole? It has to go through the primary cell wall, inner to the primary cell wall, plasma membrane present, cytosol, cytoplasm and vacuole. I think third is okay. Third one is the answer for question number 12. They don't have a secondary cell wall. Root hair cells, so the root cells, epidermal cells don't have secondary cell wall, only primary cell wall. So outermost part is the primary cell wall, inner to that the cell membrane, inner to that the cytoplasm, like that. Okay. Question number 13, which of the following correctly describe the function of smooth endoplasmic reticulum? Protein synthesis, smooth endoplasmic reticulum, no. Transport materials within the cell, correct. Synthesis of glycoprotein, no. Metabolism of carbohydrate, correct. Storage of calcium ions, correct. So B, D, E. B, D, E. Question number 14, select the statement regarding all double membrane bounded organelles. Now, double membrane bounded organelles are nucleus, mitochondria, chloroplast. They contain linear DNA, no. They are structures containing ribosomes. They are asking correctly. They are structures. Nucleus, no ribosomes inside. Ribosomal subunits can be there. Ribosome production occur, but no ribosomes, complete ribosomes inside the nucleus. Metabolism of carbo, outer membrane is smooth. No, nucleus outer membrane is rough due to RER. Then membranes, intermembrane space present, correct. Then internal membrane synthesis, membrane system present, no, not in the nucleus. Fourth one is the answer. Fourth one is the answer. Wait, people don't mark anything. I cannot. Some issue is there. Can't slide this. People, my computer is stuck now. Computer stuck completely. I can't do anything. Hope you can hear me. I think I have to go for a restart. Now I have to go for a restart. I can't even see you now. It's the third time today. Can you see me? I don't think you can see me now. Can you? I can't see. Shall I go for a restart? I don't know. I have to. Okay. I think I recovered from that. All right. Let's try now. Okay. I don't know. This is new Zoom app. Right. Question number 15. Select the correct relationship. Select the correct relationship regarding extracellular components. Extracellular components. Cell wall. Some proteins have thin flexible cell wall. Correct. Second cell wall is a structure containing cellulose. Correct. Plasmodesmata form pores in adjacent plant cell walls. Correct. Extracellular matrix contain glycoproteins. Correct. Cell junction join neighboring plasma membrane wrong. Only thing is you have not learned this yet. So I'll teach you. It's actually connecting people the cytoplasms of the neighboring cells. Right? Plasmodesmata form pores in the adjacent plant cell wall, correct. So I'm taking fifth one as the answer for that question. I will move to the structured part. Let's move to the structured part now. Don't annotate. No need. I'll peacefully I'll write now. Right. What is life fever? Here to say special unique. What do you call? Special unique state that cannot be explained using laws of physics and chemistry. That can be explained with laws of physics and chemistry. Right. Second one, what is meant by heredity? Heredity. Now here go according to the resource book. We don't have. Is there any simple definition given for heredity? We can say that acquiring of parental characters for organisms, but that is not given. Boys, guys, what is the simple definition given? We can see you. Good, good. What is the definition given? Heredity? They have genes that pass from one generation to next and control specific physiological, morphological, and behavioral characters of organisms. This is the transferring of specific physiological, morphological, and behavioral characters from parents to progenies. We'll take it like that, right? Transferring of specific morphological, physiological, and behavioral characters from one generation to another. Okay, well, that's better. From one generation to another. That is better. Okay? From one generation to another. The next one, what is the major property of water that helps organisms to cool their body surface? What are the people? High heat of vaporization. High heat of vaporization. High heat of vaporization. And then what is the monomer unit present in hemicellulose? Pentose and hexoses. Pentoses and hexoses. Next one, draw the way of the formation of peptide bond. OH group of one amino acid. OH group of one amino acid and hydrogen from another amino acid forms water molecule. Draw the way. Oh my God. Yeah, I'm wrong. It says draw. Okay, you can't write that. Oops, I'm wrong. Let me fix it. Nitrogen. I think it's better to draw the product also, and then label the product, right? So we'll try to manage space. We'll take it like this. We'll show it like this. Fine. This is better. Okay. Next one. Next one. Write two functions of mRNA. What are you going to write as two functions of mRNA? You can say copy genetic information stored in DNA. Stored in DNA. Molecule has specific sequence of nitrogen base. Right. Second one is the transport genetic information from nuclear plasm to the site of protein synthesis. Transport genetic information from nucleoplasm to site of protein synthesis. Right, next one, name two hydrophobic molecules present in plasma membrane. People, name two hydrophobic molecules present in plasma membrane. Now you can write all those three lipid types. We'll take that the hydrocarbon tails of phospholipids. Glycolipids. and cholesterol. Hydrophobic molecules present. Now we know we have hydrophilic and hydrophobic both properties present in membrane constituents. So even you can't take proteins. They also have hydrophilic and hydrophobic regions. Name two hydrophobic molecules present in plasma membrane. Fully hydrophobic molecules are not available. So I have a question. I'm going to leave that. Phospholipids and cholesterol. Right. So I'll leave that as a question. What are the main features of plasma? So is glycoprotein hydrophobic? No, we can't say that. You can't say that, right? Because the carbohydrate containing parts are actually hydrophilic. Can't we write phospholipids and no? Phospholipids are amphipathic, ne? Phospholipids are amphipathic. Polar and unpolar, both features are there. So I don't think we have a suitable answer here. What are main features of plasma membrane? What are main features of plasma membrane? I'll take 7 nanometers thickness. What else? You can see phospholipid bilayer. It is plasmodesmata. What is that? Ayyo. Plasmodesmata. Okay, we didn't discuss this part, ne? I read it as plasma membrane. Okay, so we'll just leave that. Plasmodesmata we will discuss in the class. These are cytoplasmic threads passing through the cell wall and middle lamella. Threads lined by cell membrane. Pass through. Cell wall. So we'll talk about that later. What are different types of peroxisomes and glycosomes? What are two differences between peroxisomes and glycosomes? What are you going to write? Peroxisomes. Peroxisomes. They can they have enzymes to break down H2O2. Ne? We'll take that, people. They contain enzymes to break down enzymes to break down H2O2. They are absent in glycosome. Then you can say that the peroxisomes present in plants and animal cells. Glycosomes in plant cells. Only in plant cells. Right? You can take some features like that. And next one, state importance of enzymes present in membrane. State importance of enzymes present in membrane. We learned that, people, some of these membrane enzymes are helping in food digestion. Right? They are present in microvilli and all. Helps in food digestion. helps in food digestion. What are organelles? What do you say, people? Subcellular components. Subcellular components of the cell bounded by membrane. Bounded by membrane. Then what are average diameter of the nucleus? What is the average diameter of the nucleus? We can take 5 micrometers. State main components of the nucleus. What are you going to write? Nuclear envelope. Second one, you can take nuclear matrix. Third one, you can take nucleolus. Last one, you can take chromatin. Chromatin. Then question number 5, state 2 functions of nucleus other than synthesis of different materials. Control cellular activities. And then what else will store and transport genetic information. Store and transport genetic information. Right. Then state difference between ER and Golgi apparatus. What are you going to see? ER Golgi difference? ER flattened or tubular sacs? ER flattened. O tubular sacs. But Golgi flattened. What else you can write? You can say people ER what else you write? No lysosome. ER can't form lysosomes. Golgi form lysosomes. ER has some ER has neither. ER continues with the nucleus. Golgi connected with the ER, right? Golgi. Then we'll take that ER does not carry out storage. ER synthesize lipids, oils, steroids, plasma membrane materials. But then Golgi forms cell wall materials, cellulose and non-cellulose materials. We even discuss people, one is light microscopic, the other one is electron microscopic, like that. But we cannot write them because the resource book does not give any information like that. Continues with outer membrane of the nuclear lock. Continue with outer membrane. Of nucleus. This is ER. And lysosome, sorry, Golgi is not. Golgi is not. Cyst phase, trans phase present neither. right? We'll see that cis phase, trans phase neither. No cis, trans phase. Here, cis, trans, present. You can take like that. Okay, now people, for the essay question, I didn't prepare a marker scheme today. Ultra structure means electron microscopic structure. You have to draw the diagram, right? EM diagram you have to draw for the mitochondrial EM diagram, and then there's a small description given. You have to write that. Chloroplast also you have to draw that electron microscopic weave of the chloroplast, and then there's a small description given. You have to write that. Right? I think you can do it. There is nothing to right plan for that. Just draw the diagram, and then draw the diagram, and then people write the description. Okay. So that's all for the day. Right? I'm stuck once again. I'm stuck. Right? Again, I'm stuck. Okay, then people, I'll meet you some other time. Meet you some other time. Right. Right. I'm going to finish this. Okay, bye.